Hello, everyone. So this talk is a little bit unusual. It comes with a warning. When I first presented it at Write Speak Code, they were like, maybe you should have, just some of the visuals are, I just can't put my finger on it, but they're weird. And they are weird. We're going to look at some weird things, and we're going to talk about some unsettling things. And all sorts of things tend to like pour downriver when you open dams like that. And so if you can't be here for it, then please take care of yourself. But if you can, and I hope you can, welcome. My name's Ashi, and I'm a senior software engineer at GitHub, which has nothing whatsoever to do with this talk, because today I'm going to talk about a hobby of mine. I want to share with you some of the things I've learned at the intersection of computational neuroscience and artificial intelligence. So for many years, I've been fascinated by how we think, how we perceive, how it is that the machineries of our brain and bodies go and create qualitative experiences. Goodbye, screen. This is just going to be a talking talk. There are no visuals in this talk.
There's a panel behind the TV there. You just hit the button. This happened left.
Okay. I also love how I'm standing so it'll come down on my head.
Should we know?
Oh.
This will be bloopers by Ashi.
I mean, who puts a who puts a tablet on something that pops it out and then drops it on the floor?
We like check on the ground. That's what this will be.
Yeah. I mean, they did say eject panel, so I mean, that's what I asked for, I guess. Oh, good. I didn't have to like repair and reset up my displays. All right. So, where was I? I was being fascinated by how I think and why our experiences are shaped like they are and why we suffer, why we design things like that. And I've also been fascinated by AI, and I think we all kind of have. We're watching these software systems begin to approximate the tasks of our cognition in ways that are sometimes downright unsettling. And so today, I want to share with you some of what I've learned. Some of it is very solid research, and some of it is solid speculation. And all of it speaks to a truth that I have come to believe, which is that we are computations, are worlds created on an ancient computer, powerful beyond imagining. So let's begin. Part 1, hallucinations. This person is Miquel Preo Nieto, and he has something to show us. It's going to start with these simple patterns, splotches of light and dark, like images from the first eyes. These will give way to more complex shapes, to lines and colors, and then curves and hatches. We are diving through the layers of the Inception image classifier, and it seems like there are whole worlds in here. These shaded, multichromatic hatches, crystalline farm fields of an alien world, the cells of plants. To understand where these visuals are coming from, let's take a look inside. The job of an image classifier is to reshape its input, which is a square of pixels, into its output, which is a probability distribution. The probability that this picture contains a cat, the probability of a dog, a banana, a toaster. It performs the reshaping through a series of convolutional filters. Now, convolutional filters are basically Photoshop filters. They're made of neurons, and each neuron in a convolutional layer has a receptive field. That's some small patch of the previous layer that it's looking at. Each convolutional layer applies a filter. Specifically, it applies an image kernel just a square of numbers, where each number represents the weight of the corresponding input neuron. And so we multiply the input neuron's values by those weights, and we sum them all up, and we get the output value. Now, this same filter is applied for all neurons across the layer, and the filter is learned during training. So the way training works is we feed the classifier a labeled image, something where it knows what's in it, and it outputs predictions, and then we math to figure out how wrong those predictions were. And then we math again, nudging each and every single filter in the direction that would have produced a better result. The term for this is gradient descent. Now, to make these trippy deep dream visuals, we invert that process. So the visualization we see here is recursive. To compute the next frame, we feed the previous frame into the network, and we run it through the network's many layers until we get to a layer that we're interested in. And then we math. What could we do to the input image to make it stimulate this layer more? And then we nudge the pixels of the input image in that direction. The term for this is gradient ascent. Finally, we scale the image up very slightly before feeding it back into the network to generate the next frame. This keeps the process from just enhancing the same details in the same place over and over again. And it also creates this really wild zooming effect. Every 100 frames or so, we move to a deeper layer, or a layer slightly off to the side. The network we're using is Inception, and Inception has quite a lot of layers, and they're not quite arranged in a neat linear stack. And that gives us this. Where we started with these rudiments of light and shadow, deeper, we find like a city of Kodama's situation happening. And then shortly we will enter the spider observation zone, where spiders observe you. It's okay, because the spiders are going to become corgis, and the corgis are going to become the 70s. Deeper, there's the space of nearly human eyes, which will become dog slugs and dog slug birds. Down here, there was an unfortunate saxophonist teleporter accident. And finally, the flesh zones with a side of lizard. So when I first saw this layer, I thought to myself, speaking it to no one, that it looked like Donald Trump. And I swore I would never say anything, and then I showed my best friend, and she was like, you know. And I think that says more about the state of our neural networks than this one, and I think the lizard juxtaposition has something to do with it. But I do want you to look at and notice and think about what it means that all of the flesh in this network is really white. So this is pretty trippy. Why is that? What does it mean for something to be trippy? To figure that out, let's take a look inside ourselves. Meet Scully. Scully doesn't need all this cruft, because we're just looking at Scully's visual system, our visual system, which starts in our retinas. Now, our retinas are pretty weird. The light comes into them, and it immediately hits this membrane. There's a layer of ganglions, which are not generally photoreceptive, although some of them are a little. They're neurons that will eventually relay the signals from the photoreceptors to the rest of our brain. There's another layer of tissue that does important stuff. I don't know, it's not that important. And then at the very back are the photoreceptors. Now, these are the rods and cones. They sense color and luminance. So when light comes into our eyes, it has to wind its way through 3 layers of tissue and then eventually hit a photoreceptor. That photoreceptor is going to get excited. It's going to send out a signal to the ganglions, which have to send it to our brain. And they have to do that by sending it through the optic nerve, which goes through a hole in the center of our eye. You can actually see this blind spot if you do some visual tricks. So our image sensors are mounted backwards, and there's a hole drilled through the center of them. And we don't notice any of this because we patch it all up later in software. There's another problem here, which is that if you figure out the bandwidth of a single neuron, and you figure out how many neurons there are on the optic nerve, you'll see that the optic nerve has about 10 megabits of bandwidth. And I don't know if you've ever tried using like video conferencing over a 10 megabit connection. It's like much slower than the slowest Wi-Fi. And I think our vision is better than that. My vision seems better than that generally. And how does that happen? The way that happens is that our retinas compress the data. So each of these ganglions is connected to a receptive field, some subset of all the photoreceptors. When its entire field is illuminated, all the ganglions fire weakly. When the outer part of its receptive field is illuminated and the center part is dark, half of the ganglions in your retina fire strongly, and the other half don't fire at all. And then the other half of your ganglions behave in the opposite way. They fire strongly when the center is bright and the surround is dark. So if you kind of reason this out and think about it, what this is doing is it's constructing an edge detection filter. The strongest pulses occur where there is a transition from light to dark. And this lets us downsample the image in our retinas 100 times while still retaining some vitally important information, namely where the edges of objects are. So this encoded signal then goes through the brain. It hits the optic chiasma, where the data streams from your left and right eyes cross, giving you stereo 3D vision. It's processed by the thalamus, which is responsible, amongst other things, for running your eye's autofocus, which is something that you have, something that until I read about it, I had never thought about. So every step of the way, we're extracting some information from this signal. The thalamus also processes auditory signals, and so there's some crossover there. And all of that is before we get around to the main visual processing center, the visual cortex, which is in the back. So our visual cortex is arranged into a stack of neuronal layers. The signal stays pretty spatially oriented. Oh no. Oh well. That photo's not there. The signal stays pretty spatially oriented through each of these layers. So there's some slice of tissue in the back of your brain that is responsible for pulling faces out of one particular chunk of your visual field, with all of the redundancies and slop that we've come to expect from any neural network, artificial or biological. Each neuron in a layer of our visual cortex has a receptive field, which is some chunk of the previous layer that it's looking at. And neurons in a given layer respond the same way to stimulus in their receptive field. And the way that they respond tends to extract different features from the signal. So first, our brains extract low-level features like lines and shapes and edges and colors, and then more complex features like gradients and surfaces, objects, motion, eyes, and faces. So that's very similar to the behavior we see in Inception, and that's no accident. Convolutional neural networks like Inception were inspired by the structure of our visual cortex. Now, our visual cortex is also different from Inception in many ways. Where Inception is a straight shot through, our visual cortex contains feedback loops, these pyramidal neurons that connect deeper layers to earlier ones. Those feedback loops allow the results of deeper layers to inform the behavior of earlier ones. And so we might say turn up the gain for edge detection where we have later detected an object. So this process lets our brains focus, not optically, but attentionally, well before we become consciously aware of it, improving our visual predictions over time. And I think you know this feeling. It's the feeling of thinking you see one thing and then realizing it's something else. So the loopback pyramidal cells in our visual cortex are covered in serotonin receptors. Different kinds of pyramidal cells respond to serotonin differently, but generally, they find it exciting. And don't we all? You might be familiar with serotonin from its starring role as the target of typical antidepressants, which tend to be serotonin reuptake inhibitors. When serotonin gets released into the brain, they make it stick around longer, thereby treating depression. Some side effects may occur. Most serotonin in our bodies is actually located in our gut, where it controls bowel movement. It signals to our gut to go on and do what it does to food. And that seems to be what the molecule signals throughout our body: resource availability. And for animals with complex societies like ours, those resources can be very abstract, social resources, as well as energetic ones. That your pyramidal cells respond excitedly to serotonin suggests that we focus on that which we believe will nourish us. Now, it's not correct as a blanket statement to say that the pyramidal cells in our brain are excited by serotonin. In fact, there are different kinds of receptors, and their binding tends to produce different effects. So there's 5-HT1A receptors, which tend to be somewhat inhibitory. It can cause a kind of drowsiness. 5-HT3 receptors in the brain are associated with the sensation of queasiness and anxiety. And in the gut, they make it run as well, only backwards. So a lot of antiemetics and anti-nausea drugs are 5-HT3 inhibitors. There's another serotonin receptor, and it's one that pyramidal cells in our brain find particularly exciting. Oh, thank God, that came up. This is the 5-HT2A receptor. It is the primary target for every known psychedelic drug. It is what enables us to have psychedelic experiences. So you go to a show, and you eat a little piece of paper, and that piece of paper makes its way down to your stomach, where it dissolves, releasing molecules of LSD into your gut. Now, lysergic acid diethylamide does not bind to 5-HT3 receptors particularly, and so if you're feeling butterflies in your stomach, it's probably just because you're excited for what's going to happen. And what's going to happen is this: the LSD will diffuse into your blood. It will cross the blood-brain barrier. It has no trouble doing so. It's a tiny molecule, but powerful, like you. It will diffuse deep into your brain, into your visual cortex, where it finds a pyramidal 5-HT2A receptor and locks into place. The LSD molecule stays bound for around 221 minutes. It's 4 hours, which is an astonishingly long time. They think that a couple of proteins snap into place and form a lid over top of the receptor, trapping the LSD inside, which would help explain why LSD is so very potent, with typical doses about 1,000 times smaller than most other drugs. And so while it's rattling around in there, the little molecule of LSD is simulating a feedback loop in our visual cortex. It is sending the signal that says, pay attention. Whatever you're looking at, it may be nourishing, it's important. And so the pattern-finding machinery in our cortex starts to run overtime and at different rates. In one moment, the pattern in a tapestry seems to extend into the world beyond it. In another, it's the trees that are growing and breathing. The perception of movement, a visual hypothesis that's been allowed to grow wild. With Deep Dream, we asked what would excite some layer of Inception, and then we adjusted the input image in that direction. But there's no comparable gradient ascent process in the biological psychedelic experience. And that's because we aren't looking at a source image. We are looking at the output of the network. We are the output of the network. The output of your visual cortex is a signal carrying visual perceptions, something like qualia, which will be integrated by other circuits in your brain to produce your next moment of conscious experience. Inception never gets that far. We never ask it what it sees in all this. But we could. We could perform the amplification process on a final result rather than an intermediate one. Maybe we could ask, what would it take for you to see this banana as a toaster? Or say, don't these skiers kind of look like a dog? These are adversarial examples. They are images tuned to give classifiers frank hallucinations, the confident belief that they are seeing something that just isn't there. And they're not totally wild, these robot delusions. I mean, that sticker really does look like a toaster. It's very shiny, too. And those skiers, I mean, kind of look like a dog. Like you can kind of see where the head would be and then where the tail, they don't actually look like that dog. But if someone were tired and far away and drunk, they might for a moment think that they were looking at a big dog. But they wouldn't conclude that they're looking at a big dog. And that's because the recurrent properties of our visual cortex, not to mention the whole rest of our brain, give us a stateful, continuously shifting view of the world. It is a continuously refined hypothesis, the state of which is held in the state of our neurons, which may actually point the way towards more robust recognition architectures, recurrent convolutional neural networks that ruminate upon images, making better classifications, or providing a signal that something is wrong. Because there are adversarial examples for our visual system, and they feel very weird to look at. No. Oh, we're not looking at them, though. Some of the images didn't transfer to my new computer after I poured kombucha on the old one. Sorry, old computer. When we look at optical illusions, though, and you'll have to close your eyes and imagine, you know the feeling. You know the feeling of your perception of the scene flipping between many alternatives. You know the feeling of looking at something, a pair of things which you know are the same color, and yet which just don't appear to be. There's something scintillating in it. It's like we can feel the machinery of our brains flipping and changing. And if we designed convolutional neural networks with recurrence, they could exhibit such behavior as well, which maybe doesn't sound like such a good thing. I mean, do we want image classifiers that are vacillating and uncertain? Do we want them driving our cars, for example? But we drive our cars. And it's our ability to hem and haw and reconsider our own perceptions at many levels that gives our perceptual system such tremendous robustness. So paradoxically, being able to second guess ourselves allows us greater confidence in our predictions. We're doing science in every moment, the cells of our brain continuously reconsidering and refining a shifting hypothesis about the state of the world, which gives us the ability to adapt and operate in a pretty wild range of conditions, even while we're tripping face, or while we're asleep. Part 2, dreams. So these are not real people. These are photos of fake celebrities dreamt up by a generative adversarial network, a pair of networks which are particularly creative. The networks get better through continuous mutual refinement, and it works like this. On the one side, we have the creator. And this is a deep learning network not unlike Inception, but trained to run in reverse. This network we feed with noise, just a random bunch of numbers, and it learns to generate images. But how does it learn to generate images from noise? It has no way to learn the game. In the technical parlance, it lacks a gradient without another opponent, without the adversary. Now, the adversary is an image classifier, like Inception, but trained on only 2 classes, real and fake. Its job is to distinguish the creator's forgeries from true faces. And so this network we feed with ground truth, with actual examples of the faces of celebrities, and the adversary learns. And then we use those results to train the creator. If it makes a satisfying forgery, it's doing well. If its forgery is detected, we backpropagate the failure so that it can learn. So I should tell you that the technical terms for these networks are the generator and the discriminator. I changed the names because names are important and powerful and meaningless. They don't change the structure of the training methodology, which is that of a game, a semi-supervised learning technique. We haven't found all of the images that this generator could ever generate and labeled them plausible and implausible. That would be an impossible task. Instead, we have this process of recursive co-training, where we have two neural circuits that together ruminate on a space of possibilities, extracting significant value from a relatively small amount of training data. A process that might be helpful for neural circuits of all kinds, though it does have some quirks. Meet Fallout Cow. Generative adversarial networks are not especially good at global structure. And so here we have a cow that has grown an extra body, just as you have perhaps spent a night walking through a house that is your house, but with many extra rooms. And these networks are also not extremely good at counting. So this monkey has 8 eyes because sometimes science goes too far. Just do something for me. Next time you think you're awake, which I think is now, count your fingers, just to be sure. Now, if you have more or fewer than you expected, try not to wake up just yet, because we're not quite done. Another interesting thing about GANs is that the generator is being fed noise, a vector of noise, literally some point in a very high-dimensional space. And it learns a mapping from that space onto its generation target, in this case, faces. And if we take the point in that space and just kind of drag it around, we get this, which is also really trippy. This resembles things that I've seen, that someone who isn't me has seen on acid. This resembles things that you might have seen in an old dream. Now, I don't have a magic school bus ride to take us on to understand why that is, but I do have a theory. And the theory is that when we see a face, there's a bunch of neurons that light up in our brain, and together they start resonating the signal that is the feeling of looking at that face. Taken together, all of the neurons involved in face detection produce a vector embedding, a mapping from faces to positions in this high-dimensional space of cognitive states. And as we're dragging around the generator's vector, we're also dragging around our own, which is a novel and unsettling sensation. So that's a kind of wild theory, but it's not totally without neurocognitive precedent. Here we have a rat in a cage. We've hooked up an electrode to one part of the rat's brain, to a single neuron, and those pink dots are the locations where it fires. If we speed this up, then we're going to see a pattern start to emerge. This neuron is a grid cell, so named because the centers of its firing fields form a triangular grid. Now, there's lots of grid cells in your brain, or in rats' brains, but probably in yours as well. And each one aligns to a slightly different grid. They collect data from your visual system and from head direction cells, which encode the direction of your head. And together, they seem to construct an encoding of our position in 2D Euclidean space. This operates even in our sleep. If earlier you discovered that you're dreaming, and you want to see the end of this talk, but you're not quite sure how to stay in it, maybe you're starting to wake up. Oneironauts, people who lucid dream for a living, sleeping, they recommend spinning around here in the dream, which detaches your perceived body, that is the one with 12 fingers and several extra bedrooms, from your physical body, which is lying in bed. This positioning system is something which on some level you always knew existed. You have a sense of space. You know where you are. You know when you are moving. And it's likely, even necessary, if we believe that cognition is computation, that our qualitative sense of position has a neurocognitive precursor, some signal in the web that tells us where we're at, in many senses of the word. Part 3. Sticks and stones. Now they say you can't tickle yourself because you know it's coming. Specifically, when your brain sends an action command to your muscles, that's called an efference. When an efference is sent, your brain makes a copy. Now, makes a copy sounds very planned. It sounds like someone wrote code and engineered this. Your brain is this big, messy, evolved signal processing mesh. So another way to think of efference copies is as reflections. And we take the efference and we send it out to our peripheral nerves, where presumably it's going to make some muscles contract. Meanwhile, from the efference copy, we predict how our body's state will change. And we use that to update our brain's model of our body's state. If we didn't do that, then we would have to wait for sensory data to come back to tell us what had happened. Where is my hand right now? And then we would face the same problem as trying to play a twitchy video game over a crap connection. Signals take about 10 milliseconds to go from your brain to your hands, and another 10 milliseconds to come back. It's not that low latency or high bandwidth, our nervous system. And 20 milliseconds of latency might be fine if you're playing Quake or Fortnite, but it's not good enough for moving body parts in a smooth, coordinated way. And so in order to do that, our brain has to make predictions. So life goes on. But in a moment, we're going to have a problem because we still receive sense data from our nerves. If we update our models again, they'll actually fall out of sync, as though I moved my fingers twice. And so this signal we know to attenuate so that the model stays in sync. This attenuation seems to apply to our sense of touch as well, when that touch is an expected consequence of our movement. Now, this model is obviously fairly complicated, and aspects of it are probably distributed throughout our brain. But there's one place that seems particularly important in maintaining it: the cerebellum. Cerebellum is quite special. All action commands from the brain to the body route through it. It contains about half of the neurons in our entire nervous system. Every sensation from the body to the brain goes through it as well. It has long been recognized as vitally important to motor coordination. So, something like this. People with cerebellar damage have trouble performing movements like that smoothly. With cerebellar damage, our movements become jerky, laggy. Now, it's theorized that the cerebellum acts as a Smith predictor, our brain's controller for our latency-distant bodies, able to estimate the body's current state, integrate sensory feedback in order to update that model, and decompose gross actions generated elsewhere in the brain into a fine-tuned, continuously varying control signal. And once you've evolved it, a thing like that has a lot of uses. There's a growing body of evidence implicating the cerebellum in language, which does make sense. Utterance is a kind of movement. And language, she said, kind of gesticulating wildly, is not limited to utterance. The work of moving words is not so different from the work of moving the body. They are both transformations from the space of internal states, of efference and ideas, to the space of world coordinates and external sense impressions and other people, and then back again. And what happens when this predictor encounters a problem? When there's an irreconcilable discontinuity in the model? Damn. So these things we do, they're not so different. They're visceral, they're guttural, they shake our bones. Jokes are kind of shaped like trauma. They are both shatterings. They're both the illuminations of paradoxes and discontinuities, things which cannot be and yet are. Things which we must revisit again and again, churning water, trying to smooth the edges of cutting stone, the machinery of our brains trying to make sense of a world that resists it. Preparing this talk was a difficult time for me. I didn't know if I could do it. The world is falling apart. And I got dumped, which definitely didn't help. There were days when I would open my email and every subject line was a stone, and I thought to put them all in my dress and wade out into the sea, but I didn't. Because I remembered that I am a process of creation. I am a song, singing myself. We are stories telling ourselves. We are the sea understanding itself, our churning waves creating every moment of exquisite joy and exquisite agony and everything else. It's you. It's all you. You are everything. Everything you have ever seen, and every place you have ever been, and every song you have ever sung, and every God you have ever prayed to, and every person you have ever loved, and the boundaries between you and them, and the sea and the stars, are all in your head. Thank you.